So, welcome to Biology Practical 203, which was paper 2, and this paper was supposed to take 2.5 hours. So this is the way the paper was marked. I've already given a video of the requirements. So in that video, you know what is C, D, L, all right? And therefore, the first thing in equation 1 is that you are told you are provided with specimens labeled C, D, and a solution labeled L. State the habitat of specimen C. The habitat of specimen C was aquatic. So you can say aquatic, or you could have said water, or fresh water. But if you wrote sea water, it was rejected. All right. Name the trophic level occupied by specimen C. And the trophic level is producer, or, this is ecology. You say first level or first trophic level. This is a practical testing the topic ecology. Reject primary producer, one level, okay? And there is no need to qualify. All right? So the answer should be producer, first level, or first trophic level, not primary producer. Give a reason for your answer in 2 above. The answer which you are supposed to have given was, it has chlorophyll for photosynthesis. All right? You can see for photosynthesis is bracketed. So you could have just said it has chlorophyll. Okay? You could alternatively say it is green, or it is green, it has green coloring matter. Those ones could have earned you marked. You could also accept, or the examiners could also accept, where if you wrote the reason as chlorophyllus. So if you wrote the one chlorophyllus, you are given. Note this, A1 and A2 are tied. So if you fail A1, you cannot get A2. That's the meaning of tying. B. Place 5 cubic centimeters of solution L into a 100 ml beaker. Using a straw, blow gently into the solution. Record your observation. And the examiners expected you to say this. Color changes to yellow. Color changes to green. Or color changes yellowish green. Or color changes greenish yellow. Or color changes orange. And these color differences are as a result of the amount of carbon dioxide present that time. So it depends on the amount of carbon dioxide, or CO2. Explain the observation in C1 above. Carbon dioxide in exhaled air contains carbon 4 oxide. Okay? Oh, sorry. Let me repeat. The observation is that carbon 4 oxide in exhaled air. So bracket means you can omit that. Or the other one was exhaled air contains carbon 4 oxide. Or you could just have said CO2 and you get the mark. Reject wrong formula. Remember, CO2 starts with a capital letter and O is also a capital letter. B1 and B2 are tied. So you cannot fail B1 and you get B2. Examine specimen D using hand lens. Giving a reason, state the division to which the specimen belongs. And if you did that one, you found that the division is mycophyta. And also, those who said mycota got the mark. Reject spelling mistake of the word mycophyta. It must be correct. In biology, you call something what it is. You don't imagine and start giving the wrong spellings. Even missing a letter is the wrong spelling. Also reject mycophyta or fungi. All right? And also reject fungi alone. So mycophyta, and you bracket fungi, you spoil the answer. We just need mycophyta. But if they give fungi alone, you can mark the reasons. Examine specimen D using a hand reason. So what is the reason why it belongs to mycophyta? It is non-green, all right? Or no chlorophyll, or has hyphae, all right? So any of those alternatives are correct. What role is played by specimen D in an ecosystem? Specimen D is a decomposer. Or if you don't remember that one, you can say it causes decay or rotting or breaking down organic matter, or you say decomposition, or you say putrefaction. Reject the role as if they say the role is for recycling nutrients. You put a very big wrong. What role is played by specimen D in an ecosystem? Is a decomposer, or it causes decay or rotting, breaking down organic matter, decomposition, putrefaction. I think this one we have repeated the question. I beg your pardon. So it's more like re-emphasizing. Reject recycling of nutrients. Draw and label D, 4 marks. All right. So we draw and label D. Let me try to put this one closer so that we see whether we can see details more without even seeing the word drawing D. We are dealing with D now. Yes. You are marking as follows. There was drawing mark 1 and drawing mark 2. All right. Drawing mark 1, at least 2 vertical hyphae with sporangium correctly drawn, except double line or single line. Mark D2, the whole diagram must have a continuous outline and no shading. So they have given alternative diagrams there, but you can see the diagram there, and you can see where we were, the D1 drawing mark was being put. D1 there against the hyphae, D2 at the stern. So the rebels are sporangium, hyphae or vertical hyphae, or reproductive hyphae, or if you say sporangiophora, all those were correct. And then there was stern. So those are the 3 rebel marks. So how did you award the marks? Drawing was 2 marks. Rebel was 3 marks. Maximum 2 marks. When you add them, you get the total 4 marks. Question 2. You are provided with specimen labeled E 0.01% DCPIP and 0.1% ascorbic acid. Examine specimen E. What part of a plant is specimen E? The answer was fruit. Give a reason for your answer in 1 above. It is a fruit because it has 2 scars. Or if you don't remember that, you can say it has a point of attachment to receptacle and the remains of the style. A1 is tied to A2. Draw and label one of the cut surfaces, and you can see what we have done there. We have drawn. So let me focus on 1. Drawing. So let's push this one here. We focus on that one there. So you can see we have drawing mark 1 and drawing mark 2 and drawing mark 3. Drawing mark 1, double line for epicap, continuous outline. Drawing mark 2, which is there, at the corner there, seeds located, seeds should have a loculi and juice sac in at least 1 loculi to get that mark. D3, centrally placed placenta enclosed in a ring. Centrally placed placenta enclosed in a ring. That's why you get D3. So each diagram mark has got the conditions as I've said. D1, double outline for epicap, continuous outline. D2, seeds must be present, loculi must be present, and juice sac in at least 1 loculi. And then D3, you place the placenta at the center. All right. So let's look at the rebel mark. So the rebel marks are epicup, or you can call it exocup. All right. Then we have the mesocup. All right. Then we have the endocup. And then we have the seed and the placenta. Guys. Kata transfer section through specimen E. State the magnification of your drawing. The magnification is between 3 and 5. So you write it as x3 or x4 or x5. State the type of presentation of specimen E. The type of presentation is axial. Accept axo or central, but reject axial or free central. Look at the spellings very carefully, guys. Name the agent of dispersal of specimen E. Animal. Accept man or human beings. Reject human without human beings, or reject other specific animals. State how specimen E is adapted to its mode of dispersal. So here we have, the first one is that seeds have hand slippery coat to prevent digestion. Seeds have hand coat to prevent digestion, yes, or seeds have slimy coat to prevent digestion, or seeds have slippery coat to prevent digestion. Those are three alternatives. I beg your pardon there. It is scented to attract the agent, or aromatic to attract the agent, or sweet smelling to attract the agent. Number 3, succulent to attract agent, or succulent, hence edible by the agent. Juicy, hence edible by the agent. Freshy, hence edible by the agent. Brightly colored to attract the agent of dispersal. Adaptation must be qualified. That is, if you say succulent, you have to say to attract the agent. You cannot just say succulent as their mode of dispersal or the adaptation for dispersal and then omit attract. Okay? Hand, coat to prevent digestion. So you have to say to prevent digestion. You cannot just say hand coat. Fine. To 1 cubic centimeter of DCPIP in test tube and 1% solution of ascorbic acid, drop by drop until the color of DCPIP disappears. Shake the test tube after addition of each drop. Record the number of drops used. So the number of drops is 1 to 4 drops. For example, 2 drops or 3 drops, 4 drops, or 1 drop. But you cannot say 1 to 4. You have to be specific. The ones you added, not guesswork. Squeeze out the juice from specimen E into a beaker, filter, and discard the residue. All right. To another 1 cubic centimeter of DCIP in a test tube and the juice from specimen E drop by drop. Shake the test tube after addition of each drop until the color of the DCPIP disappears. Record the number of drops. All right, record the number of drops. All right. used, so it is 1 to 10 drops in G8. So if you say 1 drop, sawa. If you say 2 drops, okay. 3 drops, but you cannot give the range at 1 to 10. No. State the specific number. From the results obtained in C1 and 2 above, calculate the percentage of ascorbic acid in the juice obtained from specimen E, 2 marks. Show you are working. So there you have drops of ascorbic acid used, drops of juice used, times 0.1%. So that one will be equal to 2/8 times 0.1%. All right? And that one gives you to 0.025%. Ensure the division is correct. Units percentage must be included in the answer. State 2 factors that influence the accuracy of the result. Size of the drops or size of dropper, and then presence of impurities. Suggest the expected results if the juice from the specimen E was boiled for 30 minutes, cooled, added drop by drop to DCPIP solution, no color change of DCPIP, or blue color of DCPIP remains, or DCPIP would not be decolorized. Explain the expected results in F1 above. Okay. Boiling destroys ascorbic acid, or heating destroys ascorbic acid, or high temperatures destroys ascorbic acid. Accept destroy ascorbic acid without mention of heating and boiling. Reject denaturing or killing because it is protein. Deny boiling or heating alone must qualify what the boiling and the heating does. Question 3. You are provided with specimen labeled B. Name the class to which the specimen belongs. The specimen is pieces. Deny spelling mistake of technical term pieces. Deny if they write pieces and in brackets they put fish, but continue marking the reasons. Give 2 reasons for your answer in A1 above. Presence of scales, fins, gills, operculum, lateral line. Mark first 2. A1 and A2 are tied. You are provided with specimen labeled B. What term is used to describe the shape of the specimen? Streamlined. Accept streamline. Stroke the specimen from the head to the tail. Record your observation. From the head to the tail, smooth, or you say slippery. Tail towards the head. Record your observation. Rough. Okay, stroking from tail to the head, rough. Stroking from the head to the tail, smooth. What is the significance of your observation in C1 above and 2 above? It reduces friction or resistance during locomotion. Measure in millimeters the length of the specimen from the tip of the mouth to the tail. The measurement of the length is 50 to 300 millimeters depending on the size of the fish. And the tail from the anus to the tip of the tail mark is 21 millimeters to 150 depending with the size of the ship. So any measurement between those is correct. Using the measurements in D1 and 2, above calculated the tail power, reading in D1 over reading in D2 times 100%, which is going to be 21 over 150 times 100%. So the tail power is 42%, or 150 times 300 on the biggest times 100% equals 50%. So the answer lies between 42% and 50%. Name and draw the fins of specimen, enable the specimen to balance and break direction. Those are the fins there: pectoral fin and pelvic fin. Okay, let me take this one closer for the fins. Deny if more than 2 fins are named. Deny a whole fish with all the fins. Accept a whole fish if the 2 fins are labeled correctly. Reject if 3 fins identified. Drawing mark. Spelling of the name must be correct. Pectoral fin, pelvic fin. Correct shape, continuous outline. Correct origin of spines. Majority of spines must touch the base, except single line spine. So you get 4 marks. Drawing 1. And the other drawing there, 1. So that you have 4 marks. Guys, that is the end of me taking you through the way Biology Practical 2003 paper was marked. Remember, I was active participant in the marking. Bye-bye, guys, and sleep well. Have a good night. Watch my videos throughout, and don't forget to subscribe to my channel for more videos like this.